So thank you very much, Anna Paula. Thank you for the organizer to inviting me. As a medical entomologist, I try to give a kind of different perspective on the topics that we are all interested in. So this is the basic vectorial capacity McDonald formula, which summarizes the most important parameters that are affecting the vectorial capacity of a species or a vectorial system. And here we are focusing on exotic arbovirus like dengue, chikungunya, Zika, etc. Very quickly, because the previous speaker already introduced my presentation, so I'm very thankful. I just want to remind you there are two main vectors of exotic tropical arbovirus, Aedes aegypti and Aedes albopictus. They are both very aggressive and diurnal biters, and they are container breeders, and this allow them to adapt from small natural breeding size. Oops, it's very fantastic this, but I don't know how to use. So very fantastic, very natural breeding size to very artificial ones, which increase the species synanthropism, so association with humans, and also allow them to become very highly invasive, thanks to oviposition on eggs in new styles that were transmitted all over the world, and in the last 30, 40 years allow the species to spread worldwide. However, there are also big differences between the two species. Aedes aegypti is still a largely tropical range. Is very highly anthropophagic. It means that bites humans even if other hosts are available. It bites mostly in south houses, and is for these reasons is a primary vector of tropical arboviruses in tropical areas. Albopictus became also capable to invade temperate region. This is why we have it now in Europe, because it produces eggs that are adapted to overwinter, and has a lower anthropophagy compared to aegypti, lower tendency of entering into the houses, and for this reason is a secondary vector arbovirus in the tropics, but still is our only primary vector in Europe. And because I do this, why I am doing this comparison? Because sometimes, because we don't know many things, many details on Aedes albopictus biology and life cycles, we tend to assume that they are the same of aegypti, but they really don't are. And also the other thing is that how can I go back? Okay, yes, okay. The other thing that I would like to anticipate and to point out is that these parameters that you can see here are highly affected by climate, of course, and also ecological conditions. So what we know about Aedes albopictus in its native range cannot be assumed to be true for its invasive range. So the biology and the bionomics and the phonology of the species in Europe. So among all these variables parameter, the vector competence and the extrinsic incubation period are parameters that can be assessed in the lab. So I'm not going to say that are easy, but there are standard approaches that can be implemented in few selected labs with safety structure, and which can allow to address temperature variations and to identify the temperature ranges of temperate climates in relation to these parameters. But these are studies that are possible, but are still limited in numbers because are complicated. But even more complicated are studies addressing vector density, vector mating rate, vector survival, because these are studies that have to be carried out in the fields. Protocols and are not always available, And these parameters are very sensible to both climate and ecological conditions. And these are the parameters on which I'm going to focus my presentation a little bit more. So how, as entomologists, can we assess these parameters? I'm going to show a few examples of our work in the medical entomological group of the Department of Public Health and Infectious Disease in Sapienza University. Was too long. Okay, so first of all, mosquito density, or mosquito counts, or relative abundance. It seems, okay, no, fine, thank you. There are, of course, approaches to collect mosquitoes. We can collect, I don't understand how these things work. Okay, yes, so we can collect adult mosquitoes. The gold standard is by collection on human volunteers, which of course it's the most effective, but is operator dependent, is very laborious, and is unethical when there is virus transmission. We also have host taking traps that in a way try to simulate the presence of hosts and to collect mosquitoes. But the most used trap to monitor mosquito Aedes albopictus density are ovitraps. Are traps of small water containers that were a wooden stick is inserted, and the mosquitoes lay eggs on the sticks, and we count the eggs. So it's very cheap, very easy to implement on large scale, and it's largely used in Italy, but also in Europe to monitor Aedes albopictus. However, our question was how we're sure that number of eggs in ovitraps reflect the number of adults in the wild. And to address this point, we did a very large study in the framework of the AIM-COST action, which was a large networking action I coordinated a few years ago, and which allowed to put together 18 teams from 13 countries, which for 6 months carried out collection, albopictus collection by ovitraps and by human landing catches. And here you can see the relationship between the mean number of eggs per site per week and the mean adults, mean number of adults per site per collections. And you can see that there is a relationship, and then there are, of course, countries where the mean abundance are much higher in both samples than others. And also the overall correlation between this set of data is quite high. So judging from this, we can say, yes, of course, so we can use eggs instead of adults in our models because they're more or less reflecting each other. But when you look at the data from single sites, you can see there are some sites where the correlation is still very strong, but there are also some other sites where you can see an opposite correlation or very large confidence interval. So the bottom line of this is that eggs are not always good predictor of adult density, and I think this is something very important. The paper is in preparation since a lot of time, but it will be published sooner or later. And I think it should be important for the research community to be aware that considering eggs as predictors of adults is not always a good idea. So another part of our group research work was to use a different kind of trap that you can see there on the bottom, sticky traps. They resemble ovitraps, but they have some internal walls lined with adhesive material. So the female mosquito, when try to lay eggs, she remains stuck to this adhesive layer, and we can count directly the adult mosquitoes. And we used this trap for several works, one of which was to study dispersal of the mosquitoes by mark release recapture methods, experiments which are quite laborious. Just to give you a short overview on how difficult these studies are. You have to collect eggs in the field, you have to rear the larvae, you have to get the adults and mark them with fluorescent dust, then you have to release, then you have to recapture them in a wide area, and then you have to count marked against the non-marked mosquitoes. And based on this, we established the flight range of these mosquitoes, which is around 200 meters, and based on which we now know that when there is an infected cases, we have to treat an area of 200 meters around the infected cases to prevent most of the risk of the transmission. But I'm not going to go into the detail of the flight range, but I just want to say that we use this data to calculate the recapture rate, the relationship between marked mosquito against non-marked mosquitoes in the experiments. We did 6 replicates in 2 sites, and the recapture rates of the sticky trap were about between 3 and 9%. And why this is important? Because this allowed to move from count of mosquitoes to estimates of absolute abundances. And we use this data to calibrate a model to determine the absolute abundance of Aedes albopictus in Rome. These are sites across Rome that have been sampled with sticky traps in 2012 and 2023. And you can see there that the absolute number of albopictus is estimated were higher in most sites, in all sites except one in 2023, and also that the length of the season was larger in 2023 than 2012. And the thing is that 2023 has been recorded at the hottest years ever. Now we have a new record, which is 2024, but at the time this was the hottest year. So this means we can establish by this work that a very hot season, as the one that we experienced last year, increase the abundance, absolute abundance of the vector, and also, which we think is very important and relates with what Flavia was saying about the epidemics, the length of the transmission of dengue, extend the season to large part of the year. This work was submitted. Chiara, who is the first author, is there, so any question about this work can be asked to Chiara. And based on this, we also estimated R0 for dengue and chikungunya, again in 2012 and 2023. And you can see that in 2003, again, the risk R0 is higher in some countries, in some areas of Rome compared to 2012. And that, again, the season is larger than it is in the former year. So now that we know the recapture rate of sticky traps, which are traps that are used almost only by our research group, we try to use this information to assess the capture rate of other kind of traps, which are more widely used by the research community, in particular BG traps, which are adult traps that attract mosquitoes by CO2 and other specific odors. I don't have time to go into the detail, but we did a kind of Latin square experiment where we tested the BG traps collection and the sticky traps collection in 4 sites across Italy, and this paper is published if you are interested. It was published this year. And the thing that we established that there is a multiplicative factor of 14.5 of BG versus sticky traps, which means that we can conclude that by any 10 females in a BG trap collected over 24-hour period corresponds more or less to 300 female per hectare. So by having this multiplicative factor defined, we can establish more or less absolute female densities, abundances based on BG traps collections. And moving to a different, I don't know how much time do I have, but 5 minutes, perfect, thank you. To more rapidly to other parameters which are important. One of these is the vector biting rate. The vector biting rate, it's also dependent from mosquito abundance, but also from host abundance, and also from different host availability, because if you remember one of my first slides, Aedes albopictus is considered not a strong anthropophagic species, so it's expected to bite also other humans, other host, other humans, and this is what we tried to establish a few years ago. So we analyzed the blood meal content of blood-fed mosquitoes captured in urban area in Rome, and you can see that 92% of the blood meal were carried out on humans, which means that in urban area we have a very, very strong biting rate of albopictus to humans. But then if you move to different environment, like for instance the cemetery, where there is a very important cat colonies, you can see that 30% of the blood meal are taken from cats. And if you move to rural area, you can see that other animals become the primary hosts, and humans are lower affected by albopictus biting. So this, I think, is something that has to be considered carefully when assessing the transmission, risk of transmission of arboviruses, because it changes. This parameter is one of the parameters that changes very significantly from urban to rural areas. And finally, a last long standing question in medical entomology in general is related to how to assess the probability of survival of the mosquito vectors, which are very, very have a big weight in the McDonald formula. The thing is that we don't have good tools to assess mosquito age, and traditionally this is done morphologically, but it's very complicated, much more competent on albopictus than on other species for some reason. So it's really not realistic to trust on this technology to assess the age structure of a mosquito population. Recently we have been working in collaboration with Glasgow University to use mid-infrared spectrometry associated to supervised machine learning to develop an algorithm and an approach to assess more easily the age of mosquito population. Just to give you again an idea how difficult is this from our perspective, to do this we had to rear mosquitoes under laboratory and semi-field condition in order to have mosquito of known ages. And then mid-infrared spectra were obtained for this mosquito of known ages. The observation were labeled. Some of them were used as training set, other as test set. There were different prediction model based on machine learning developed, and eventually this is one of the results we obtained. So we have obtained quite high accuracy in determining the age of 3 large age groups, which we define as for sure not infectious, low probability of being infection, higher probability in being infectious based on different ages. And these approaches we are working, already working on the next phase, which is not to determine the chronological age, but the biological age, which depends on the ecological conditions, and will provide eventually, if successful, a better estimate of the probability of daily survival, that what we need to establish the risk of transmission. And this is in progress. So to the take-home message already, Anna Paula anticipated this. Do we know enough about the epidemiological and biological parameter? No. It is, I hope that I convince you that it's very difficult to assess these parameters, but I believe that it's important that we as entomologists and you as modelists interact and synergize in order to also for us to understand which are the most important parameters that are needed, and for you, for your models, and for us also to highlight the one that from our perspective may have a major impact. So having said this, I thank all the research group, in particular Beniamino Caputo, who is sitting there, with whom I co-lead all our research activities, and also our collaboration with the Foundation MAC that since a lot of years opened to us the wonderful world of models, and Fondazione Buto Cluster, which more recent times became very strong collaborators. So thank you very much.